So here you see two protein structures. I'm focusing on the protein structure evolution. The left one is the CO dehydrogenase, this one. And the other one is not familiar to them, to you. It's called hybrid cluster protein, which is evolutionarily related to CODH. First of all, I will introduce a brief information about CODH. CODH catalyzes the CO2 conversion to CO and the reverse reaction at the several conditions. The overall structure is normally a homodimeric here, and there are 5 metal clusters here, 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 here. The most important metal cluster is called C cluster here, which shows the chemical structures like this one. And the names of the metal clusters are here, as shown here, C, C, B, D, B, because some of the CODH types can interact with acetyl-CoA synthase, which contains A cluster. So please keep in mind this structure, because today's main topic is a hybrid cluster protein, which has a similar cluster. Also, the C cluster reaction mechanism can be intensively studied. So this red box figure is a resting state, the starting point for the catalysis. First of all, the CO2 molecule is attached to the nickel ion to make a nickel carbon bond, organometallic bond. Then normally the CO2 molecule is a linear structure, but once this is bound to the nickel side, this is a bent form. After that, the CO2 molecule is further attacked by the OH minus moiety here on the Fe1 side to here, or then this moiety could be released as a water. Then the rest of the CO moiety is formed on the nickel center. Then the CO is released. So this mechanism is rarely studied. Furthermore, we briefly look at the diversity of the CODH enzymes. This figure is a little bit old, but it's a good summary. In this organism, there are 5 classes of CODH enzymes. And the most famous CODH class is the CODH3, which can be associated with acetyl-CoA synthase here as ACS gene product. But you can see several other CODH enzymes, gene products, here, which also can function as a CODH reaction, CO conversion to CO2. So that means in chemistry viewpoint, CO-CO2 conversion, interconversion, is found in most of all CODH types, except for CODH5. And the reaction is reversible. Some bioinformaticians have also studied the relationships of the CODH based on the primary amino acid structures. And here is one example. The nickel CODH, this group formed a big cradle here, and the typical CODH enzymes, CODH class 2, is located in this position. CODH5, QS5, is a little bit unique CODH, and it's a bit far from this normal CODH. And now you see this one, HCP grade. So there are several, somehow relationship between HCP and CODH, but only about 15 to 20% amino acid sequence similarity, identity, could be observed between two types of enzymes. So it's clear these two enzymes could function in different manner, but the question is how the protein structure of HCP. Actually, in about 20 years ago, one class of HCP has been studied by structural biology. Actually, this HCP, class 1 HCP, is a monomeric enzyme, and it contains 2 different types of metal clusters. One is here not typical, typical for sulfur. The other is here, which is the catalytic center. This enzyme function in a different way from the CODH. It can convert NH2OH to NH3 or NO to N2O. But the detailed mechanism of the CODH-HCPs are unclarified yet. The active site structure has been already reported, but it's clear there are big difference because this cluster has no nickel. Instead, some of the oxygen atoms or oxygen species could be observed in this manner. But you can imagine, you can remember this the left side of this cluster, the architecture in this side, the architecture of the cluster shape, it's like a kind of a little bit cubic, is similar to the C cluster in CODH. Now we can compare two HEP and CODH overall structures and the active site structures. I would like to note that HEP is monomeric, but the yellow colored area is a unique region for only HEP. And this region is not observed, no, HEP, not CODH. Active site structure, metal clusters are like here. So this site are similar. And you can see some of the amino acid residues coordinating to the metals are common. But the others, for example, system sulfide is different, not observed in this site. So we are curious about how the reactivity of this cluster and how this cluster is formed inside the cells. But many of the things are unclear at this moment. We've just started the study of HCP. And classification of HCPs is also important to think of the evolutionary history of HCPs and CODHs. Actually, there are three different classes of HCPs as like CODHs. Class 1 is a monomeric, and the structure is solved, and mainly found in anaerobic microorganisms, bacteria, archaea, and even unicellular prokaryotes. Class 2 is found only in facultative anaerobe, such as E. coli. And one unique feature is this class utilizes the HCP reductase called HCR as a specific partner. Class 3 is rather much unique one because this forms are homodimeric like CODH. It's clearly different from these two classes. And this class of HCP is found anaerobic bacteria and archaea, not eukarya. So my question is, how is the structure of class 2 and class 3 HCPs? Because it's important to gain some insight to solve the structure of all of the HCPs. So our experimental setup is like this. Fortunately, E. coli has his own HCP, class 2 HCP. That means we can produce recombinant enzymes in E. coli system. But you can imagine, if you want to produce a large amount of proteins, the cofactors should also be produced a lot. Therefore, we introduced the ISC operon, which is involved in the biosynthesis of iron sulfur cluster inside the E. coli cell, together with the HCP gene. Then, after the expression of both of the gene products, we purified only HCP under the anaerobic condition using this kind of tent. The crystals of HCP, E. coli HCPs are like this. The color of this protein crystals are typical iron sulfur cluster type, brown or reddish brown. Then we solve the crystal structure of the E. coli class 2 HCP as shown in this left side, and now we can compare both of the structure of class 2 and class 1. The overall structure of both of the classes are almost identical. However, one unique feature was found in class 2, here, the cis-rich loop. The cis-rich loop is very close adjacent to the N-terminal 4-iron-4 sulfur cluster, which may suggest the redox system could be different between this one and this one. And as we've already known, this HCP specifically utilizes the HCR enzyme, unlike this protein. Therefore, this structure could be important, but we have to study. We further focus on the geometry loop structure here. The unique class 2 specific loop is forming the protrusion using proline structure here. So from this figure, from this panel, the protrusion is heading to the outside, to the solvent area. On the other hand, class 1 HCP has no protrusion, and it's like positively charged due to the lysine. The electrolytic structure on the surface are like this, and you can see the hydrophobic area is mainly observed around the N-terminal region of class 2 HCP. Instead, the class 1 HCP has more positively charged region near the 4-iron-4 cluster. This is very convincing to think of the difference in the redox partner between class 2 and class 1. Then we are curious how the redox partner can interact with the class 2 HCP, HCR. Unfortunately, the crystal structure of HCR has not been solved yet, but we can easily model the protein structure by using the homologue here, futarate dioxygenase reductase. The successful model of HCR is like this, and the conserved system motifs are localized in one domain here, which could bind iron sulfur cluster, actually 2-iron-2 sulfur cluster. In addition, the FAD or FMN binding site is located in the other domain, which could work as an electron transfer. Actually, this HCR utilizes the FAD rather than FMN, which can be analyzed by mass and EPR and so on. Here is the UVBs and EPR spectra of HCR, and one interesting feature is this signal here. This signal, which may be difficult to interpret, but the sharp signal of EPR is normally assigned to the organic radical rather than the inorganic radical. This means the electron transfer system of HCR can utilize the organic radical, which can be interpreted as FADH radical. So the reaction is between HCR with HCP using electron power is like an electron bifurcating, but it's not exactly. But now we have several materials to think of the complex structure of HCP, HCR. Then we compute the complex structure by docking estimation. And finally, we found two of meaningful complex structure, predicted structure, to explain how the electron transfer occur. Here. At this moment, we couldn't decide which is correct or which is not correct. But you see the distances between the cofactor binding site of HCR to HCP 4-iron-4 sulfur to the actinic site hyperclusters acceptable, somehow acceptable, and the route for electron transfer could be like in this way or this way. More important feature of the N-terminal region could be addressed, in my opinion. So we prepared one mutant here, which has one HCP mutant, which has a class 1 type HCP loop. The activity assay shows this mutant has no activity, and one interesting feature is EPR here. So this EPR is not popular, but very specific to detect the FEO site, and this indicates FEO center is observed in wild type, but not in the mutant. Very recently, we successfully solved the executive structure of class 3 HCP having the homodynamic architecture here. And the overall structure is very, very similar to CODH. And now we can compare all of the classes of HCP overall structures and CODH. It's clear to see this protein architecture is very similar to this one. And the other important feature is the inner and outer helices here. It's colored region. Actually, these colored helices are very, very similar. When we compare cyan and dark blue helices, the sequences of these helices are very similar. Also, this kind of similarity is found between yellow and red and orange and brown. This means these helices could be duplicated during the evolution from the class 3 HCP to class 1 or class 2 HCPs. As I told you, the class 3 HCP and CODH are similar, but there are several differences. For example, here, the interfaces between two enzymes, this HCP has hydrophobic environment. This has not hydrophilic. This is one distinct feature between two enzymes. And also, of course, we can see the active site structures are different. And some of the helices on the surface area in CODH are different. These blue colored helices are not found in HCP. And now I introduce a very recent work, not from my work, but from the other work, CODH5, which has no CODH activity, because this enzyme has FeSO type cluster like HCP, not nickel containing one. However, the cluster architecture are not completely the same, but they're different. The big difference is found the cysteine persulfide here. The cysteine persulfide is very unique, rare ligand in metal enzymes, and the property of cysteine persulfide and normal cysteine thiolate are very, very different. So at this moment, we don't know why cysteine persulfide exists in only HCP, but not in this protein. But when we intensively study the reaction mechanism of HCP, maybe cysteine persulfide role of this moiety could be solved, hopefully. Now we have structure information like this, and several CODH structures by the other group. And now we summarize the information about the HCP and CODH, especially the homodynamic architecture of HCP class 3. Then we computed the phylogenetic trees. For this experiment, we used the 3D dimensional structures superimposition. Then once the superimposition was achieved, the superimposed amino acid sequences are extracted to make this tree. Then we see class 3 is very close to the CODH clade, and CODH5 is also close to the HCP region. And typical nickel-containing CODHs are far from this HCP region. And among the HCP classes, the order of the evolution may be 3 to 1. But at this moment, we need more data to determine the evolution of this area. So now, sorry. Now we have one hypothetical scenario to understand the structural evolution of the HCP-CODH family. If the HCP class 3 is the origin of this type of family, for HCP evolution, the dimerization could be broken to monomeric one, and some of the loops are modified. For CODH evolution, first of all, the central region could be modified, especially the interfacial higher sulfur cluster. Then nickel-containing C-cluster maturation system could be obtained during the evolution. But we, of course, need to study how the genes, how the nickel-containing systems could be evolved by seeking for the genomes of some of the microorganisms. Finally, I would like to thank for the students and co-workers as shown here, and also I would like to thank for the financial support by CO World, JSPS, and Tokyo Chemical Corporation Foundation. Thank you very much.